Welcome, everyone. My name is Michael Lemke. I am the Senior Director of Science and Industry Affairs at the Alliance for Regenerative Medicine. Welcome today to the Aging webinar on regulatory considerations. This is the 6th webinar in the Aging case study series. Before I turn over to our expert speakers, I'll give you a little bit of background about Aging. This is a project that was first conceived of at an ARM CMC focused workshop in December of 2017, where our members identified the lack of best practices in manufacturing as one of the issues that was a rate limiter in the advancement of the field. Aging is a case study of the manufacture of an AAV vector using a triple transfection process and an HEK 293 cell line, applying quality by design principles. It was put together with the help of funding from NIMBLE, led by ARM, and content was contributed by ARM subject matter experts from many different organizations involved in the development and manufacture of gene therapies. And you'll hear from 2 of them today. We are currently in an educational rollout for Aging targeted at ARM members. On this link here on the slide, you can find information about upcoming webinars. I believe we have one more on downstream processing, tentatively scheduled for March. And then we have plans a little later on in the year to do a half-day workshop focusing on the entire case study. So look for more information on that soon. Aging is open source for the community. The document itself is on ARM's website. You can find it very easily in the search engine on our website or by directly following this link here on the slide. Sorry. With that, I would like to introduce the speakers for today. Saroj Ramdas is the Vice President of CMC, Regulatory and Technical Strategy at Amicus Therapeutics. Previously, she was a GSK Fellow and Director of CMC Biopharm, Regulatory Advocacy, Strategy, and Policy at GlaxoSmithKline. In her current role, she leads a team responsible for delivering robust CMC regulatory and technical strategies to support a diverse portfolio in collaboration with technical operations, quality, and global regulatory affairs. Saroj has 24 years of experience in product development, sterile manufacturing, and CMC regulatory, strategy, and advocacy. She holds an MS from Medical College of Virginia in biochemistry and a BS in chemistry from the College of William and Mary. Also speaking today will be Alex Balmer-Sassi. Dr. Balmer-Sassi is a Senior Director, CMC at Voisin Consulting Life Sciences. She has over 20 years of experience in the biopharmaceutical industry and consulting services. At BCLS, Alex is responsible for the strategy and technical content of projects involving the CMC development of biologics and cell and gene therapies. Prior to joining BCLS, she was a CMC regulatory executive at GlaxoSmithKline, where she developed CMC strategy and coordinated regulatory submissions for marketing authorization in the US, EU, Japan, and emerging markets. Dr. Balmer-Sassi received her PhD in pharmaceutical sciences from the University of Pittsburgh and her pharmacy degree from the University of Sao Paulo, Brazil. She completed her postdoctoral studies at McGee Women's Research Institute in Pittsburgh. Saroj, before I turn over to you, just a few logistical details for the webinar. There will be a Q&A session after the presentation. Please, participants, feel free to submit questions at any time. I would ask that you use the Q&A tab for that. After the webinar today, we will post the recording of the webinar and the slides to ARM's member portal. Saroj, with that, I turn over to you.
Thanks a lot, Mike. Let me share my screen. Great. Thanks, everyone, and thanks, Mike, for the opportunity to be here. Alex and I are really excited to spend the next hour here in discussing the aging regulatory considerations and the work that was put into the chapter as well. For today's chat, we'll focus on introduction on the regulatory framework and some of the expedited pathways that are utilized within the US, EU, and Japan. I'll hand it over to Alex, who'll walk us through some of the regulatory interactions as well. And then we will touch on some case studies at a high level, which weren't included in the chapter, but I think can provide some flavor on some of the expectations on topics you could engage with regulators and some feedback at a high level as well. And we'll wrap it up with lifecycle management, just touch on that, some of the opportunities for the future in this phase, and our conclusions and acknowledgements. So I'll kick it off with the regulatory framework. Within the chapter, we have a lot of detailed information, but what I just wanted to highlight here is touch on each of the markets. And so for the US, our gene therapies are regulated by FDA's OTAT, the Office of Tissue and Advanced Therapies. And this is located within the Center for Biologics Evaluation and Research, as we all know as CBER. We've seen a lot of, in recent years, a lot of implementation of various guidance with relation to gene therapies and even in the CMC space. And some of those are highlighted by the guidance on CMC information for INDs. There's a testing for retroviral vector-based human gene therapy products, devices used with regenerative medicines and advanced therapies as well, and microbial vectors used for gene therapy, and potency tests for cellular and gene therapy are some of the ones highlighted here that are very relevant from the CMC space. In addition, the FDA has a really nice educational resource for industry, and they've established this support via OTAT, and the link is found here on the slides, but I would really encourage folks on the call here to take a look at some of the content they have within the link and the resource established by OTAT. On the left, we have highlighted some of those guidelines as well, and links for these can be found in the chapter. Secondly, I'll touch on the EU. So in the EU, gene therapy products are included under the umbrella of the HCMPs, and these are regulated by the EMA's Committee for Advanced Therapies, or CAT. And CAT has a multifunctional role within the review process. It just certifies quality and non-clinical data for small and medium enterprises developing HCMPs. It does assist in scientifically developing documentation related to the regulations, so our guidances. It contributes to scientific advice and supports the scientific advice working party as well. And it also trains assessors and organizes scientific workshops as well. And within the EU, we've also seen activity in this space with guidance that have been put in place regarding quality, non-clinical, and clinical aspects for gene therapy products. There's the scientific requirements for environmental risk assessment of gene therapy products as well, and also the quality, non-clinical, and clinical aspects of medicinal products containing genetically modified cells. And then there is some guidance on marketing authorization procedures as well that could be really useful for members on the call. And we can move into the regulatory framework with Japan. Japan has two main regulatory authorities that are independent agencies with really distinct roles. You have the Ministry of Health, Labor, and Welfare. I think most of us know it as the MHLW, and you have the Pharmaceuticals and Medical Devices Agencies, the PMDA. So the MHLW is responsible for the guidelines, managing advisory committees, and providing final authorization. And the PMDA is really focused on the regulatory and scientific review. And for gene therapies, this occurs within the Office of Cellular and Tissue-Based Products. It's also involved with GMP compliance and inspection. I found this diagram to be really helpful in differentiating the roles and responsibilities. And if you focus on the blue, which I think is very relevant from a day-to-day perspective with the PMDA with respect to projects that will go into review, you can see the relationship of the Office of Cellular and Tissue-Based Products and the Office of Manufacturing Quality and Compliance as well. in terms of GMP oversight for manufacturing sites as well. Their novel and diverse nature of gene therapies, a key theme that we can find across all three of these regulators is it's an evolving space. Guidance is evolving, and in all three of these markets. So it's really important for us to just keep an eye on what's coming up in the pipeline from a guidance perspective as well. I'll switch gears to talk through what are some of our expedited pathways within each of these markets. Many of us are probably very familiar with these from other modalities, but these are also available, these pathways are available within the gene therapy space. So within the US and CBER, you have two pathways that you could utilize. You have the breakthrough designation. This was established in 2012. And some key features of this program would be early and more frequent communications with the FDA during development. You can have a rolling submission and review as well, and you could also have a priority review. BTD is really helpful in terms of a partnership with the agency to help define some of your development strategies in looking to accelerate to get to the clinic. In addition to BTD, you have RMAT, the Regenerative Medicine Advanced Therapy within the US. And this was established in 2017 and has the similar benefits of BTD, but the additional piece here is the opportunity for early discussion on potential surrogates or intermediate clinical endpoints. So not only can you engage from a CMC perspective to help accelerate development, but there's a potential opportunity here to have an accelerated clinical outcome as well. Within PRIME is established in the EU, and Alex will be going into more detail in some of these pathways as well. This was established in 2016, and this also allows that enhanced interaction and early communication with sponsors. You have the opportunity for accelerated assessment as well as ongoing scientific advice through your development. And in the last column, we've highlighted Sakigake. This is an accelerated expedited pathway in Japan, which was established in 2016. This pathway affords you prioritized consultation and review. There's also substantial post-marketing safety measures that would be discussed, and this allows a rolling submission and review process. For the Sakigake process, another piece would be that you would need to be submitting in Japan around the same time as you would say in the US and EU, or this product would need to have been developed in Japan. So it's just an additional criteria there. So what is this? What do these pathways afford us from an industry perspective? Some of the benefits here would be really the early engagement and hopefully often engagement you can have and get some regulator feedback to de-risk items that could put you on clinical hold. The other piece would be the feedback informing your product development strategy. So if you engage earlier, you're able to then get some feedback that will define what that product development strategy is. And what that can then lead to is an accelerated development and initiation of your clinical studies as well. I'll hand it over to Alex to walk us through the regulatory interactions.
Thanks, Arosh. So I'll cover the regulatory interaction with the health authority, and I will start with the FDA. So throughout the development of the gene therapy product, right, you have several opportunities for interactions with FDA from early phase with the Interact meeting, then a pre-IND meeting, and then opportunities during the clinical development and prior to product registration. Now, the Interact meeting is a quite interesting one, as the sponsors can obtain preliminary non-binding feedback from the agency. And this is a non-mandatory meeting, but it's still highly recommended. Now, the challenge for this type of meeting is really to identify the right time to approach the agency, because it can be too soon and the meeting would be denied because you don't have enough information, or you're a bit too late and the meeting is denied because the FDA will see that you're ready for a pre-IND meeting instead. Now, the type of questions throughout those meetings, they will change as the product development progresses. For example, again, in an Interact meeting, you may be asking about the testing strategy. for the release and characterization without being ready to share a defined acceptance criteria. And then when you're reaching a pre-IND meeting, then providing that acceptance criteria will be beneficial, even if you're providing a broad range. It would still be better than not providing one. So you can have some FDA feedback and avoid potential clinical holds. As you progress into later development phases, right, those questions and company positions will become more complex, always with the ultimate goal of a successful BLA submission and approval. And we'll provide a couple of case studies towards the end of the presentation. If you go in the next slides, please, Roj. So it's exciting to be in this field of cell and gene therapy because it's clear that it's in the forefront of the health authorities, and from the FDA perspective, it's definitely on the FDA's thinking, as clearly as was shown in the upcoming PDUFA 7 commitment letter. So this covers the fiscal years of 2023 through 2027. And the PDUFA commitment is really a product of FDA discussions with the regulated industry and the public stakeholders. And there are several goals planned that will benefit the development of gene therapy products, and I'll highlight some of them here. So one of them is splitting the real-time application review pilot program. So this would really allow for the submission to be done in a split fashion, where you could submit in approximately 2 months apart. Excuse me. So the goal here would be to shorten the time from the date of complete submission and allow earlier patient access to therapies that address an unmet medical need, which is basically almost all the cell and gene therapy products. In meeting management goals, there is an FDA proposal to create a type D meeting that would allow for a quick discussion on a narrow set of issues between the FDA and the sponsor. So this could be a follow-up question that raises after you have a formal meeting with the agency. There is also commitments on enhancing regulatory science and expedite development. So the FDA is aiming to enhance the communication between FDA and the sponsor, continuing with the early consultations, and ensuring that you continue the success obtained with breakthrough therapy program. There are efforts on advancing development of drugs for rare disease. So in this case, the FDA is proposing a pilot program to support efficacy endpoint development for drugs that treat rare disease conditions and offering additional engagement opportunities with the agency. Also, efforts on advanced development for drug device and combination products. This is something that we're seeing more and more in the gene delivery space, and using a device for a more effective delivery. And then the advancing of real-world evidence for decision-making. So the FDA is again looking to a pilot program for using RWE to improve the quality and acceptability of those RWE-based approaches. Next, please. Additional commitments are enhancing regulatory decision tools. This would be with a more focus on the patient, use of benefit risk assessment, drug development tools for qualification, pathway for biomarkers, model-informed approach, and innovative clinical trial designs. There is a commitment for enhancement related to product quality reviews. So the FDA is proposing several enhancements on that area, which one of them being promoting the use of structured information requests. And I think from the CMC regulatory perspective, that's going to be quite beneficial. There's a new pilot program to facilitate the expedited CMC development of products under an IND when you anticipate a significant clinical benefit. And as we are working this area, this is very aligned to what we do on a daily basis. There is an accelerated need for CMC development to meet the clinical timeline. So this can be quite interesting for us. And additional commitment on holding a public workshop to help advance the and advanced utilization and implementation of innovative manufacturing, and that can be beneficial. And last on this PDUFA is the enhancing CBER capacity. And by now we have heard several presentations from Peter Marks and others at the FDA on the challenges related to the amount of work on the FDA side. So it's good to see this commitment coming up. Now, those commitments are aligned with the thinking from the EMA side perspective, also in terms of being more patient-centric development, additional support that's being raised for HCMPs and RWE. So next, please. So this diagram summarizes the current interactions with the health authorities in Europe. And similarly to the FDA, there are several points of interaction with the EMA as well. And this can benefit the sponsor throughout the life cycle, right, of the cell and gene therapy product. So I will start with the HCMP classification and the innovation task force meetings, and I'll discuss a little bit more on those in the next couple of slides. We have opportunities prior to start with the first in humans to seek a national or EMA scientific advice. And I'll also present a little bit more on advantages and disadvantages of going into each route. And in preparation for all those interactions, in a similar manner that you would prepare for the FDA, you're looking to product-specific questions, right? The complexity of those questions will increase as you progress with your clinical development. The next slide, please. So more details on the HCMP classification. This is a classification procedure that's non-mandatory. It can be requested any time during the development up to the MAA submission. Although it's not mandatory, it's highly recommended, especially for borderline products that includes those where the classification of a cell therapy or tissue engineer product is a bit questionable. The application for this is free. And there are specific EMA templates for the submission of the request form and the briefing document. Now it's expected that the sponsor will include the mode of action and intended indication here. The recommendation is issued within 60 days. And this is not really a direct meeting with the EMA unless the EMA feels that there's a clarification needed, they will reach out. In the next one, we have the Innovation Task Force, ITF meeting. This is also a non-mandatory meeting. It's a free submission, can be performed at any time. Sponsors typically use this as an entry point to the EMA. This used to be a very high-level meeting where the EMA would provide more of a regulatory roadmap or identify the procedures that you would be eligible for without a lot of discussion on the scientific points. But in our recent experience, we have seen the EMA a bit more open in engaging with the developers via the ITF and providing feedback on the scientific content. There's still a non-binding feedback, and it will be on the development questions and the regulatory strategy. There's no formal advice letter that you would receive, and the ITF team, they may comment on the company issued minutes. On the next slide, we have the priority medicine, the PRIME scheme. As Sarosh mentioned earlier, this is early and enhanced support really to optimize the development of medicine for targeting unmet medical needs. And the overall intention is really to expedite the access of those medicines to patients. So sponsors must be eligible and they must meet some eligibility criteria that includes a few items such as potential to address those unmet medical needs, show compelling non-clinical data in a relevant model that would provide early evidence of promising activity. You have to show that first in human indicated adequate exposure for the desired pharmacotherapeutic effect and tolerability. So with that, not every sponsor is ready for a submission. And as you can see in the figure, 72% of the submissions are denied. This is a free, non-mandatory submission, and the procedure can take about 30 days after validation of the submission. So the next slide, please. So then we have the scientific advice, right? And then there are always questions on where to go first, going through a national or EMA scientific advice. With the EMA, an advantage would be that there's a high level of expertise within the EMA, and if you follow that advice, you have better chance to have a successful marketing authorization. Now, you may not always get a meeting, and once you seek the EMA scientific advice, there are some national agencies that may refuse to provide advice because you already had an EMA. So you wanted to take that into consideration. Now, another advantage of the EMA scientific advice is that you can have and actually should have multiple EMAs, scientific advice, but a disadvantage would be that there are only 8 consultations offered per year, and they follow a predetermined schedule. So you need to prepare in advance. For a national scientific advice, there are open discussions often, but not lately, of course, for obvious reasons. The face-to-face meeting was happening before. This can facilitate a future clinical trial application. But some agencies, on the other hand, can be difficult to grant meetings. The national scientific advice input, they can be really an asset for a sponsor that could utilize that feedback from the national scientific advice to consolidate and then propose in a briefing book a meeting with the EMA for a scientific advice. Now, there are also the situation which would be a disadvantage where not all national agencies are aligned or aligned with the EMA. So overall, really the decision is on a case-by-case basis, and it will depend on the strategy that you have for your clinical development, which would include the countries that you're selecting to start the trials. The next slide. So as you get close to the marketing authorization, and actually our recommendations that you sometimes go even before the phase 3, it's important to start engaging with the health technology assessment bodies. So from the EMA side, you'll be getting advice on the efficacy and safety and the recommendation on the market authorization on the European level. The HTA bodies, they'll focus more on the value for money. This is where really the discussions on pricing and reimbursement will occur on a national level. There is an opportunity for a joint EMA HTA body consultation, where the HTA body participation is on a voluntary basis. Unfortunately, there is a limited number of consultations offered per year, a very extensive list of eligibility criteria to meet. And although it's very attractive to have, it's not that easy to achieve. On the next slide, please. So on the topic of joint consultations, there is also the thinking about consultations across the Atlantic. So there's an opportunity for EMA and FDA parallel scientific advice. And this would be under the confidentiality agreement between the European Commission, the EMA, and the FDA. Now, this parallel approach really provides the EMA assessors and the FDA reviewers a way to exchange with the sponsor their views, but it does not guarantee that the outcome will be harmonized. This is a non-mandatory procedure, very limited. The timelines follow the 7th-day timeline on the scientific advice working party, and with similar to the 60 days for the Type B meeting at the FDA. This program has been offered for a few years, but in our experience, it has been very challenging for the EMA and the FDA to grant the meeting. Now, recently, I think it was last week, the FDA announced that they will be presenting a webinar on a 5-year review of the program, and the webinar is scheduled for March 16. And I'm definitely interested to learn their input on best practice and recommendations when considering a parallel scientific advice. So the link to the webinar is included in the slides. So I'll pass back to you, Saroj.
Thanks, Alex. So just touching on some of the requirements for Japan with the PMDA, I wanted to highlight some of the difference here. But the key takeaway that I'll share as the framework is really recommend having early engagement with the PMDA to map out what needs to be delivered as part of the package and when it does. But one of the items here that we wanted to highlight is the Cartagena Act that was put in place within Japan for implementation of the Cartagena Protocol, which is really regulating the living modified organisms and impact on the biological diversity. So what does that mean from a regulatory perspective? Within the pre-IND space, you would need to have a detailed risk assessment of materials of biological origin, and in addition, a consultation to assess if the asset falls under the Cartagena Act as well. And with this, you will also have an ALAMO application, and this is to obtain the approval and confirmation for use of your living modified organisms within the Cartagena Act as well. Something that's different with Japan than the US and EU, though a CMC module is not registered, you can submit a redacted M3 if you'd like, but there's no expectation to keep this information updated in terms of your release of material. If you do submit the CMC information, there is a possibility that you could get questions from the PMDA post IND submission as well. So I'll hand it back to Alex. We're going to walk through a couple of case studies and then wrap up with life cycle management.
All right. So our first case study is Interact meeting. And of course, this has been highly anonymized, but those are real case studies. This company X was developing a AAV gene therapy for a rare genetic disease indication. In the meeting, the company X was interested in receiving feedback from the FDA on the adequacy of the rat model, the route of administration, the biomarkers as potential endpoints in the clinical trial, and the use of engineering batches in non-clinical studies. And lastly, the potency assay design. It took about 4 months between the meeting request and the FDA feedback. And this was over a year ago. We're now experiencing much more shorter timelines between the request and the feedback from the agency. Overall, the feedback was positive and very useful and provided enough guidance for this company to continue on the development, which is now on the pathway for a pre-IND meeting. And I'll focus on the feedback provided on the bolded topics, which are the engineering batch and the potency assay. So for the engineering batches, they were looking into obtaining agreement from the agents to use the engineering batch for the tox studies, and then using the same process that we're going to do for the GMP batch to be used in the clinic. Now, in principle, the FDA agreed with the strategy. However, it was very early and neither lot release specs nor batch data had been provided in the briefing package. So it was unclear for the agency how the two materials would be comparable. So the FDA provided quite significant input and clear remarks on what was expected to be presented in a pre-IND meeting, so they could provide a firmer feedback on the comparability of the engineering and GMP batches. Now, on the potency assay, the company X used the approach of having the potency assay as characterization only, while the method was still being developed. Now, the FDA did not agree and recommend the sponsor to set a broad criteria based on available data and include as part of the release. The agency also suggests that the sponsor would describe the assay in more details in the pre-IND meeting package and justify the use so the FDA could provide a more targeted feedback on the question. So in the end, the Interact really served as an excellent guidance for the pre-IND meeting. In the next case study, this was end of phase 2 FDA meeting for AAV gene therapy product where the company was asking the FDA on a few points for their after their phase 2, which was the pivotal trial. So the key CMC questions on this meeting, they were related to comparability from clinical to commercial batches, concurrent validation, specs for starting materials, and the cell-based potency assay. So in the next slide. So in the comparability aspect, the FDA was unable to agree with the approach. And this was not only related to the limited number of batches that were manufactured at that point, but more importantly, the lack of information on the validation status of the analytical methods used for comparability. There were some methods that were being transferred from the CMO manufacturing the clinical batch to the CMO manufacturing the commercial batch that was not clear on how that tech transfer for the methods was going to occur. And then there was questions on the ability to demonstrate consistency on the manufacturing. So the comparability approach could not be, there was no green light from the agency on that approach. On the concurrent validation, the sponsor was taking on a bold strategy, aiming to obtain FDA agreement on presenting one batch for the PPQ in the BLA and 2 batches after the submission. So the FDA provided feedback, and it was very clear that the only way to consider that approach and to agree would be if the sponsor could provide a comprehensive testing for in-process testing, characterization, the control strategy, and data on development batches at small and full commercial scale before the FDA could agree. On the starting materials, and this was a specific question on the plasmid specification, the FDA requested the sponsor would provide all the sequencing reports and annotated sequence and any discrepancies that would be noted compared to the reference sequence. And on the cell-based potency assay, the FDA recommended that the responsiveness of the cells to the products need to be investigated under various conditions of passage, density, and the days in culture. In addition to genetic and phenotypic stability of the cells should be assessed. So in addition to that, the FDA recommended considering orthogonal methods for this potency assay. I'll pass back to you, Saroj.
So the third case study is with Company X, and this is for an AAV gene therapy product as well. This was in this company an in-licensed product and had a Type C meeting with the as a pre-pivotal meeting. But background here included that initially the company had hoped to take a pathway of the parallel scientific advice pathway. And when applied for that, they were asked to, they were denied a parallel scientific advice meeting, but were asked to separate it into 2 separate meetings. And this was the feedback from the US as well. Some of the key questions that were proposed was a proposal on comparability of the device that's used in the non-clinical study and what's being proposed for the clinical study. There was also a question on qualification and validation timing for the analytical assays to support the pivotal study, as well as the cell-based potency assay. So from the results or the feedback from this engagement regarding the comparability of the device, so non-clinical to clinical, the agency did tentatively agree regarding the device and procedure used for non-clinical may be used to support the proposed clinical trial. They did have additional feedback of compatibility for the device that you would want to assess from in the clinical space prior to the clinical space as well. With respect to the qualification and validation of our analytical assays, the company the agency agreed with the plan to qualify the analytical methods prior to the release of the product and start of the clinical study, except for the AAV ddPCR assay, which would need to be validated prior to the start of the clinical study. So this was really helpful for the company in their planning as well. And then with the in vitro potency assay and matrix, the agency agreed that the assay measurement is relevant to the mechanism of action for their product. One of the assays they did indicate must be quantitative with sufficient precision as well. So this was a matrix approach to the potency assay. So we wanted to just wrap up with life cycle management with ICH-Q12 being reaching step 4 within ICH and countries assessing implementation for step 5. It provides an opportunity for us to evaluate some of the tools even within the gene therapy space. And one of the drivers for Q12 is to really help progress innovation within this space. And you can imagine how quickly we're innovating within the cell and gene therapy space today. And so some of these tools could be very helpful. The toolkit provides a couple of opportunities. One would be a categorization of these post-approval changes to have defined pathways, either prior approval, a notification, which is moderate and low, or an internally managed by industry within your PQS. It provides for harmonization across the various regulatory authorities as well to define their current practices within these pathways. The second tool would be the post-approval change management protocol. This is a tool that's available in the EU and the US, but through Q12, this will become broadly available across other regions as well. And this would be a 2-step process to define the change strategy as step 1, and you would be reporting results in step 2. Many times you have a potential to downgrade the reporting category. The third tool would be defining established conditions. This would be established conditions or legally binding information and really depends on our product and process understanding. What we should really think about within the gene therapy space, it is a younger modality from a knowledge and understanding perspective if you compare it to small molecules or even well-characterized bioforms. But I think the approach that we should be looking to leverage here really is the platform approach. How are we generating platform knowledge and how can we leverage that in our understanding of our products as well and defining established conditions? And this tool also will clearly define what's considered supporting information within your regulatory filings, which can then help streamline some of those post-approval changes in the future. And a new concept from a regulatory perspective is the product lifecycle management document. This is a regulatory document that would capture your established conditions with the reporting categories proposed, as well as any HCMPs you've submitted. So this currently would reside in your 3.2R section and will be a living document throughout the life cycle of your product. So in conclusion, I wanted to wrap up here. Really, there's development efforts and regulatory guidelines within the gene therapy space are evolving. And I probably will repeat that a couple of times, but that's a key message that we'd like to get across. There are several opportunities to interact with the health authorities from very early in your development cycle and prior to your pre-pivotal or registration filing. Expedited regulatory pathways facilitate the ability of industry to really meet the need for bringing gene therapy to patients, putting our patient focus first. And additional approaches from health authorities are currently being evaluated. And as Alex shared with PDUFA 7, a lot of opportunities in that space, if they come to fruition, will provide a lot of benefit to industry. And so I would say we're continuously evolving within this global regulatory landscape and see that I think about gene therapies similar to probably where we were with monoclonals 25 years ago in terms of the maturity. So really look forward to progress within this modality. And we'd like to wrap up with thanking Jancy and Mike from ARM for this opportunity. as well as Emmanuel and Erica from Voice and Consulting, and Mina from Amicus, who helped with input into the slide deck, and Keiko from GSK. And a huge thanks to the large group of industry collaborators on the AGENE chapter for regulatory considerations, who are part of developing content and chapter reviews as well. So we'll open it up for questions. I'll hand it back to Mike.
Thank you both for that outstanding presentation. And I also wanted to thank you again for lending your time and expertise to the AGENE project. So we do have some questions from the audience. If audience members have additional questions, please feel free to enter those now into the Q&A tab. First question is for Alex. And one of the earlier slides, you explained the right time for the Interact meeting. And the question is, could you explain that again? And there's a specific example here. For example, should this be before or after testing the product in mice, non-clinical testing?
Yeah, I can elaborate a little bit more. It's really hard to find the Goldilocks approach, right, for the Interact meeting. You want to get there when you already have your candidate selected. That's one of the requirements to go for an Interact meeting. So you can't go with multiple compounds. And with some non-clinical data would be beneficial. So if you have already completed your tox studies, or you have some preliminary evidence of efficacy, that's still okay. But if you have a full package of your non-clinical, then the FDA may guide you into a pre-IND. Hope that answer your question.
Very good. Yes, thank you. Another question, and I'll for either one of you. Are there opportunities for non-sponsors, such as technology providers, to request a meeting with the EMA? And would the ITF meeting be would be an appropriate forum for that?
I can take on that one. Yes, and the name is actually escaping me on with the EMA for technologies, but you could also go through the ITF, and the ITF team would provide you some of guidance on which pathway to follow.
Yeah, I believe in the EMA you have the innovation technology. I'll find the name and we can share it.
It's hard to keep track of all the names. And would that be like roughly analogous to the CATT
exactly
in the US, the CBER Advanced Technologies team? Okay,
yeah.
That makes sense. So another equipment or technology question. How do you think about the equipment used to manufacture these drugs, particularly equipment that is specific to a new drug, or equipment that has been developed for a new production process? And can you comment on the applicable regulatory standards?
Maybe I can start off, Alex, but this is similar to the last question. Highly recommend you engaging the innovation groups within FDA and EMA. And Japan has the innovation manufacturing working group as well. Currently, it's very much focused on continuous manufacturing, but there's opportunity there to engage as well. So the first step would be to get feedback through these technology groups within these regulatory bodies on the equipment and the process as well. With respect to regulatory standards, that would be part of that discussion, I would think, and I can maybe, Alex, if there's anything else to add there.
Yeah, there might be some nuances on the type of manufacturing equipment that you're designing, because there is an opportunity, and that has happened in the past with the FDA, that you can have that as part of a device. So some of the electroporation techniques could be as part of a device. So depending on what type of equipment you're talking about, you could go that route.
Thank you. Question from someone who read the chapter, so that's good news. People are reading it. Within the regulatory considerations chapter, one of the examples for a Type C meeting includes discussing the adequacy of facility design. are you able to expand on that at all, or the required interactions for CDMOs entering the market who are building new facilities?
Yeah, so you would have to be connected with the sponsor that will be using your facility. So then you can request a type C meeting as part of your IND, and have that discussion on the facility design. It might be not so much on what you want to include in the facility, but you would put a proposal of what your plans are and trying to get feedback on adequacy for which phase of the clinical development or the marketing authorization that you're searching for.
Okay, so the sponsor would have to be involved in those
Yeah.
discussions, obviously. There's a question from one of the case studies, I believe, regarding the ddPCR assay. Did the agency expect full ICH validation prior to starting the clinical study? And was this for phase 1 or for the pivotal trial?
So this was a pre-pivotal, and this would be a validation for pre-pivotal. They expected the full ICH validation work to be completed, I believe, but not necessarily a finalized report.
Another question about starting materials. Have either of you noticed any differences between FDA and EMA in the level of control and GMP requirements for plasmids as starting materials?
There's not a lot of difference on the requirements. I think it's very, very similar.
Yeah.
Okay. It's an interesting question about the accelerated regulatory pathways. Are there advantages for requesting both breakthrough and RMAT? And is RMAT more for opportunities to discuss accelerated strategies with clinical endpoints?
Yeah,
I guess maybe I'll reword that a little bit. Go ahead. Sorry.
I was going to say that's a good question, and if you would request both, you're getting pretty much similar approaches and similar interaction with agents. So you do not need to request both. We have seen that companies sometimes they do, and that's a way to show investors about look what we have with the FDA. So yeah, I wouldn't recommend going for both. It's not needed.
Yeah. Also, there's a difference to the requirements as
Yes.
well, which is important, and that's discussed in more detail in the chapter.
Yeah. And I think what would drive that, just to add to that, would be your clinical strategy, depending on what endpoints you're looking at or surrogate endpoints, and that would really drive the pathway.
Got it. There's another question about the ddPCR assay in the case study. And it was stated on the slide that it should be validated prior to the start of the clinical study. Could you elaborate if qualifying the assay would be acceptable?
So I believe that company's intent was to qualify the assay, but the feedback, the agency did not agree with that, and they indicated it would need to be validated.
Okay. But the questions keep coming. We're gonna. It was a great presentation. Very interested audience. Have you heard about any specification for replication competent AAV required by the FDA? And the questioner is saying that they have an experience of individual reviewers requiring certain sensitivity.
Do you want to start with that, Saroj?
You know, from my perspective, I would agree with what the comment is, that we've seen some different reviewers' expectation regarding a specification, but I think, Alex, you might have more experience in this space.
Yeah, we are noticing a little bit of inconsistency. But bottom line is that really it's what you can show, and you demonstrate on the sensitivity of your assay. You demonstrate what you can control. You demonstrate the levels that you have seen in the non-clinical studies. So it's the overall package that you present to the agents that can change, persuade in one way or the other.
Okay. Another question CMC, preclinical and clinical safety data on the vector backbone are often relevant to all products on the platform. So if you have a platform of multiple products and generated with different INDs. If I want to submit the platform specific information as a standalone submission, so all platform products can be referenced against that submission. Is there a regulatory pathway for this in the US and EMA?
Yeah, so you can submit as a drug master file with the FDA. With the EMA, that's not going to be feasible. And I think that has been discussions that ARM and other stakeholders have had with the EMA before about how we can have master files with the EMA. So that could be an issue.
That is something we are working on. Question
Yes.
about the approach, the DMF approach in the US. This is my question. Would you then be able to reference that at a BLA stage? I've heard different things about that, or different approaches.
For during a BLA, referring back
Yeah.
to the DMF. Yes, you can.
Okay. Got a few more minutes, a few more questions. Considering electroporation and other delivery devices, can you share expectations of working with FDA and the EU and other regions on how device expertise is incorporated into meetings? So I guess that means the regulator's device expertise, if I'm understanding.
Yeah,
correctly.
that's how I'm trying to read. So with the FDA, we will have the CDRH involved, and that as you apply through CBER to have your discussion for your product, right? The FDA will bring CDRH into the discussion if you're providing as a combination. Would be a similar approach with the EMA as well, to have the device expertise in the meetings.
Okay, good. So we're winding down, just a few more questions, but we do have a few more minutes. The third case study, very popular. There's another question about that. The question is, was the ddPCR assay used for determining dose?
Good question. I think Alex and I will need to go back and get feedback on that one to confirm that, if you don't mind.
Okay. So yeah, what we'll do for any questions that don't get answered is we'll post follow-up on the ARM member forum along with the slides and the recording. Question about the master file reference that you made, Alex. Is this a type 5?
So this would be for the device, the manufacturing equipment.
That's for the way I'm
That's
interpreting
we
it,
talked
but
about
maybe
the
questioner could
Because
we also
clarification.
Yeah, we also talked about the vector, right?
Yeah.
Yeah,
so I'm not sure what the question is referring to.
The question came up right when we were talking about the vector, so that's the way I'm going to interpret it. Jade, if you want to clarify, just type in another.
Yeah.
Yes.
Yeah, but
the vector.
Yeah, the DMF types, they're very clear on the FDA website in terms of 1, 2, 3, 4, 5, and the requirements that are for each. Once you submit your application, if you are using a different one, the FDA will comment on that. So that's something that you can take into consideration as well. For the vector and things like that, it could still be as a type 2 because it could be an intermediate, considered an intermediate of the drug substance. And then a type 5 would be more for the equipment.
Okay. 2 questions that I'll take just in the time that we have remaining. There's a question about ACell. And yeah, we are in the final review process, editorial review process of ACell now. And our goal is to complete that sometime in the second quarter of this year. And there will be a regulatory chapter there as well, covering aspects that are more specific to the hypothetical autologous CAR T is the subject of the ACell study. So stay tuned on that. And there's a question about prime versus breakthrough RMAT application success rates. I'm pretty sure that we have that data at ARM. So we'll just make a note to post an answer to that question on the member portal as well, just as soon as I can pull that data. So with that, I want to thank both of the speakers again, and thank audience members for your attendance and all of your great questions. And I wish you all a good day.
Thank you. Bye-bye.
Bye-bye.